Hello, my name is Olivia Hunter, and today I will be discussing prokaryotic replication forks. So the start of replication always requires an origin. Here I've drawn the origin on theta replication. The origin of replication is where unwinding first occurs. For unwinding to continue, many things are required. Most important would be the DNA helicase, right here. That's responsible for unwinding the DNA double helix and by breaking the hydrogen bonds. Next is the single-stranded binding proteins. They bind to the separated strands and are responsible for keeping them separated. Next is the DNA gyrase, right here. It binds to the double helix in front of the replication fork in order to relieve tension from the unwinding. Next is the RNA primase, I've drawn here in blue. It binds next and lays down the RNA primer, which is needed for DNA synthesis to begin. The RNA primase lays down the primer, then detaches from the DNA strand until it's needed again for the lagging strand to begin synthesis again. Next is the DNA polymerase 3, this big circle right here. It attaches to the primer and synthesizes DNA to the 3' end. DNA polymerases add nucleotides from the 5' end to the 3' end, always. While these nucleotides are being laid down, phosphodiester bonds connect the added nucleotides to what was already laid down. DNA strands are anti-parallel, meaning the leading strand synthesizes continually from the 5' end to the 3' end, and the lagging strand synthesizes discontinually from the 3' end to the 5' end. So here you can see the fragments right here. These fragments are called Okazaki fragments. Next, the DNA polymerase 1 removes the RNA primer and replaces it with DNA, making the Okazaki fragments almost connected. The DNA ligase is really responsible for sealing the discontinuous Okazaki fragments to form one whole fragment. Thank you, that's it.